Okay, so reminders of a few things in regards to your final exam. January 26th, 9 o'clock in the morning in the gym. Okay, what time should you be here? 8, well, I'd be here a little before 8:45, okay, because the doors open at 8:45. You want to be able to come in here and check out where you're sitting. Okay, in front of the doors to the gym will be a whiteboard. On the whiteboard, they will have written, whatever exams are in the gym, probably be mostly Science 10 the time you guys write, and they'll say Science 10, period whatever, Mr. Coder, Science 10 AP, period 1, Mr. Coder, whatever. Okay, and they'll have all of those out there. You find your class, so Science 10, period 4, okay, and then it'll have the names that are in that row. We might take up more than one row because there's more than 30 people, or there's around 30 people in this class. Okay, so look, and it'll go alphabetical, so start of the alphabet to, somebody's name. If you're in between those two names, you sit in that row. That means you go to that row and you find your test. Okay, they'll already have your names on them. Walk up that row till you find your test and sit down. Everybody follow me here? Okay, so it's important that you do that. Don't sit down at somebody else's desk because that'll create confusion. And, well, it might work out for you, it might not. Okay, if you do really, really poorly and they do really, really well and you write each other's test, you screwed them and you got the good end of the deal, or I don't know. It just doesn't usually happen. Okay, no. Never happened because most of the time people know their own names and are capable of finding their own test. Okay, just make sure that you go there, unless of course you're writing in learning support, then you go to learning support and you're right there. But for the rest of you, go in, find your test, and sit down. Does that make sense? Okay, that's why you want to be here a little bit before 8:45 because you want to have a chance to find your name on the board so you know where to go. Okay. Remember the things you need to bring to the test. Couple of pens, couple of sharpened pencils, your calculator with a fresh set of batteries in it, okay, so that they don't die on you in the middle of the test, and that's it. That's all you need to bring. Okay, I mean you can bring a drink and some food as long as it's an exam appropriate snack. Okay, not a 500-gram bag of Doritos. Okay, the people around you will kill you. Okay, if you do that because it's really distracting. Right, so try and bring things that kind of makes sense as a snack, like a granola bar is kind of nice and quiet. Okay, an apple is kind of loud, depending on how good it is. Okay, I did have a person who peeled an orange once, and they got their fingers all sticky, and then they were like asking to go to the bathroom because their fingers kept sticking to the pages of the test. Like, think before you bring your snack. Okay, bring something that's easy and quick. Okay, that's really about it. What thing do you need to make sure you do not end up having on you? Your phone. Okay, you may not bring your phone to the test. Leave it in your locker, okay, or lock it in the car, whatever, but have it somewhere else, because if we catch you with it in the exam, whether you were using it for cheating or not, you had it in the exam and knew full well you shouldn't have it and you were cheating then. Okay, and it's just not good, right? We don't want to have to give somebody a 0 on their final, because guess what, guys, if you get 0 on the final, it doesn't matter what you had in the course going in. You get an incomplete for the course. All right, so you don't want to be in that situation. Bad. Okay, don't take the chance. Leave your phone somewhere else, and you'll get plenty of reminders. You can't bring in any electronic devices other than your calculator. Okay, that's the only electronic device you can bring in. Okay, everybody follow. You're not going to have time to read in my test anyway. It's going to take you close to 2 hours. So. Okay, all right. We'll start our exam review here. We'll do chemistry and bio today. Answer lots of questions if you got them. Okay, and then tomorrow we'll do physics, and global systems won't take long, and again, answer whatever questions you have on those two. Right, so the first thing we started out with was atomic theory. Obviously, there's 4 points to the atomic theory, and no, in the written response, there's not going to be a question that says, write out the 4 points of the atomic theory verbatim, because I'm not going to waste a written response question on a final exam on that, but there could be a multiple choice question, okay, about the 4 points of the atomic theory, or maybe there's a couple of questions, not so much about what are the points, but maybe some applications of the points, especially point number 3, okay, where chemical reactions happen when atoms are separated, joined, or rearranged. Okay, we talked about that and how that's important for how chemical reactions work and how we know a chemical reaction has occurred. We're producing new substances on the product side of the reaction, okay, stuff like that. So this is multiple choice. Okay, I'm going to be asking you about the atomic theory in the multiple choice. I'm not going to be asking you about names, people, and places and things like that, but I am going to ask you about these 4 points and maybe a couple of applications of them. All right, you don't need to write these down. You have these in your notes. Okay, this is just review. Write down the stuff I'm saying. This is what I'm going to ask you. That's what you should write down. Okay, everybody follow me there? Okay, this stuff you already have, but if I'm giving you tips and hints, okay, that's the kind of stuff you want to write down. Okay, questions about atomic theory? Right. Next thing we went over is naming. Okay, do you have to be able to name ionic and molecular compounds? Most definitely you do, okay? Because there's going to be a couple of multiple choice questions, you know, which is the proper name for this compound, or what's the proper formula for this compound, and you'll have to pick the right one. So there's going to be that. But also on the written response, when you have to write out a full reaction, you better be able to make the formulas the right way, okay? If you can't, it's going to be pretty hard to get full marks on those. Yeah. Now, I'm not going to ask you to balance compounds. I'm going to ask you what the formula is or what the name is. In the written response in terms of equations, like I'm going to say, here's a chemical reaction, balance it, right? Here's a chemical reaction, write it out in formula form and balance it, okay? Here's the reactants, predict the products and balance the reaction, yeah? Okay. All right, so quick review of naming here. Again, you don't need to write this down. You have these in your notes, okay? If we have, first step is identifying whether it's ionic or molecular, okay? How do we know if it's ionic? What will it always have in it? A metal, okay? Whereas molecular compounds always are 2 nonmetals. Okay, if we're naming an ionic compound, okay, we name the metal first, and then the nonmetal, and the nonmetal ends in "-ide", okay? We need to check for metals that might be multivalent, okay? And we can tell that because if we look in their box on the periodic table, they'll have 2 charges, or maybe more than that, right? But they'll definitely have more than 1 charge. If that's the case, what do I need to put in their name? Roman numerals, all right? So, couple of examples here to quickly review how that works, okay? Sodium iodide. Okay, does sodium have more than 1 possible charge? No, it's always +1, okay? And iodine is always minus. I don't know why there's a 2 there. That should just be NaI. That's terrible. Okay, NaI. There's something wrong with that one. All right, so when we're naming this, it's just going to be sodium iodide. Okay, and that's it. Metal, non-metal, ends in "-ide". For this one, iron. Iron does have more than 1 possible charge, so I will need a Roman numeral. Okay, it's going to be iron something, and what's this stuff? It's in the table of polyatomic ions, right? It's nitrates. All right, so this is going to be iron something nitrate. I got to figure out what. Okay, charge on nitrate is always -1, but there's how many nitrates? 2. So how many negative charges do I have? 2. All right, I only have 1 iron, so how many positive charges do I need from it? 2. So this is the iron with the 2+ charge. So what Roman numeral do I write in here? I. So iron 2 nitrates. Everybody following me there? Okay, we got to remember those, right? Second thing is we got to remember that those Roman numerals do not end up in the formula, right? That is a very common mistake that I see. Okay, people will write something like this and they'll go iron Okay, I don't need the Roman numeral in the formula. I can figure out from the formula what the charge on iron is. I only need it in the name where I can't figure it out. Okay, so Roman numerals only go in the names, not in the formulas. Okay, that happened quite a bit actually on our assignment and exam when we were in the chem unit. Sadie. No. Okay, only if you're writing it out in words, even then you wouldn't. Okay, when it's an element, it doesn't have a charge, so you wouldn't need to put anything on it. Only when it's in a compound does it need the Roman numerals. Okay, all right, is that ringing a bell for everybody? Okay. Now, if we're naming molecular compounds, we write the name in the same order that they were given to us in the formula. All right, so if I was given this, let's say Okay, P2O5. It's molecular because they're both nonmetals. Okay, I'm going to write diphosphorus pentaoxide. Right, these are the ones that need the prefixes. Okay, the ionic compounds do not need the prefixes. No, you got to know the prefixes. Okay, I expect that at this age you know the prefixes for at least 1 to 5. Okay, so mono, di, tri, tetra, penta. And actually after that, it's easy because it's the same as the shapes, right? Hexa, hepta, right? Yeah, just octa, nona, deca, yeah. Okay, all right. So we got to remember, we write the names in the same order. We change again the ending of the last thing to "ide." And then for this one here, we add the prefixes unless we're dealing with one where the first thing is a 1. All right, so for this one here, this would be dinitrogen trioxide. Okay, this would be carbon tetrabromide. Okay, I don't write mono on the first one. Okay, I would write mono if it applied to the second one, but I do not write it on the first one. Everybody good with the naming there? Okay. All right, writing formulas for ionic compounds. Got to write the symbols for both elements, and we usually write their charges at the same time. So let's say we're doing sodium sulfide, because that's what I had written there a minute ago. Okay, so we're doing sodium sulfide, so I write the formula for sodium, Na, and its charge is +1. And I write sulfide, sulfur, okay, and its charge -2. Now if I want to get the formula right, what do I do with these numbers? Swap and drop. So bring it down over here to 2. We don't write 1s, so now we're good. Na2S. All right, ringing a bell? What do I do if I get numbers I can reduce? Then I do so for ionic compounds. Molecular compounds, not the case. Okay? In ionic ones, it's always the lowest common form. All right, so we write charges, we drop and swap. Okay, we reduce if needed. All right, so let's try these two here. Magnesium nitride. All right, so formula for magnesium is Mg, and the charge on magnesium is 2+. And nitride is just N, or nitrogen, and its charge is -3. So what am I looking at here? Mg3N2, right? Right? And for this one, okay, I've got the Roman numeral in there, so iron 3 chloride, and the 3 means use which charge? The 3+. All right, so then I write iron, okay, 3+, chlorine, which is a 1-, and then I drop and swap. Okay, so I'm going to have FeCl3, not this. Okay, the Roman numeral does not end up in the formula. Okay, questions on that? Okay, for molecular formulas, they're the easiest. You just go by the prefixes. Okay, whatever the prefixes are, that's the number you write. Okay, so write the symbols. Okay, use the prefixes to get the numbers. So diphosphorus trioxide. So I've got phosphorus and oxygen. How many phosphorus do I have? 2. How many oxygens do I have? 3. Is this pretty easy? All right. What if it came out to 2 and 4? What would I do? No. Don't reduce. It's molecular. Remember, we're not trying to balance charges with molecular compounds. They're all negative. They're sharing the electrons. So whatever the numbers are, that's what the numbers are. Don't change it. Okay? Under. The lowest number. Whatever the lowest number. No. If I ended up like with something in here that was like that, okay? I would never write it this way. I would just write it like this. Yeah?
You know, you have like a minus and a plus, it like cancels out, right?
Yeah.
Is that if you had like wait, no, it doesn't. Because if you said if we reduce it, it like the plus and the minus, then they reduce, right? So
Yeah. Okay, so here's what happened in this one. Oxygen's a -2, lead's a +4, right?
Yeah.
Okay, Well, this is still a -2, and this is still a +4. Doesn't it still work out to 0? Okay. 2 times -2 is -4, plus +4 is 0, right? 4 times -2 is 8, -8, +8, it still works out. That's why you can reduce it. Your balancing charges and their ratio is always the same. Yeah. Okay, that's a good question, though. All right, and then for this one here, tricarbon hexahydride. So, C what? And how many hydrides? 6. What's hydride? Hydrogen. No, that's hydroxide. Hydride. Remember, hydrogen can be a metal or non-metal. Okay, do I reduce that one? No, it's molecular. If it's written first. If hydrogen's behaving as a non-metal, it'll always be listed second, right? But if it's first, metals are always written first. So if you have hydrogen something, then hydrogen's acting as a metal, it's ionic. Right, so all of our acids, remember we talked about acids, okay? All acids are like this, right? Whoops, HNO3, right? That's an acid. Hydrogen's first, it's a metal. Those are ionic compounds. Okay, properties of compounds. We did the lab where we identified all of the unknowns. Probably some multiple choice questions about that. Okay, I might give you a description of an unknown compound and ask you if you think it's ionic or molecular, or maybe I'll ask you outright what you think it might be from a list of possibilities. Okay, so if I say something like you have a blue crystalline solid that is highly soluble in water, conducts electricity in solution, and has a pH of 6, is it A, water, B, sugar, C, copper 2 sulfate, D, cobalt 2 nitrate? Okay, you would pick copper, right? Copper solutions are always blue. Okay, cobalt's always pink or red. Okay, nickel's always green. Okay, I don't know if I would get that specific on a final exam, but I would certainly ask you whether you think it's ionic or molecular based on its description. Okay, so we need to remember that ionic solutions will always conduct electricity. Okay, ionic solutions can be brightly colored. Okay, they are crystalline solids at room temperature. They can have extreme pHs, like all of the acids that have hydrogen in them, or all the bases that have hydroxide in them. Okay, and then for molecular, they can be any state at room temperature, solid, liquid, or gas. They have dull appearances usually. They're not usually colored. They're usually less soluble. They have low melting and boiling points, and they are less extreme in the pH range. They may be a weak acid or a weak base, but never strong in either direction. Okay. Questions on properties? That's basically one of those lessons you just kind of have to read over. Okay. And again, that's going to be multiple choice. Okay, not going to be in the written. The written's mostly skills for the chemistry unit. All right, chemical reactions. We went over 5 types. Must you be able to identify them? Yes, because how can you predict products of a reaction if you can't tell what reaction is going on? And yes, there could be some in the multiple choice that would say, here's a reaction, what type is it, A, B, C, or D? Right, so you got to be able to tell what kind it is. So for a synthesis reaction, usually that's an element and an element reacting to form a single compound. All right, and a decomposition reaction is the exact opposite, a compound that breaks down into its 2 elements. What can sometimes get produced in that kind of reaction that we have to be watchful for? Okay, let's deal with this one first. Okay, what can sometimes get produced here as elements that I have to watch for because they'll mess up my balancing if I don't catch them? Not the Roman numeral ones, but the special elements that can come as 2s, 4s, or 8s. Okay, now here's the thing. On your final exam, you have a full color periodic table where the special elements are highlighted. Okay, so I think I have yellow for everything that's a 2, and it's blue and orange, I think, for the ones that are 4 and 8. I don't remember which one, but it's labeled at the top. Okay, there's like blue, 4+, orange, 8+, yellow, 2+. Or sorry, not plus, comes as an 8, comes as an octet, or whatever. So the special elements are identified on the periodic table that you will get for the final exam. But obviously you got to be looking for them, because if this produces one of them and you don't put a 2 beside it or a 4 beside it, then you're going to get the question wrong. Okay, so that's one we got to be mindful of. Okay, if we have a combustion reaction, we'll have a compound containing carbon and hydrogen. Okay, reacting with oxygen gas, always producing water and carbon dioxide. Are there special balancing rules for these? Okay, what order do I balance in? Alphabetical order, and the even numbers, the rule of 2. If I have an even number of carbons, I start balancing with a 2. Okay, I just start with a 2 here as my balancing if the carbons are an even number. Okay, little things to remember. I mean, if you haven't started reviewing, those are things you probably haven't reminded yourself of. Okay, and then we have the 2 replacement reactions. Okay, remember that in replacement reactions, your compounds are always ionic. Okay, you'll never have a molecular compound in a replacement reaction. So if you accidentally make one when you're predicting the products of a reaction, hopefully you'll recognize you did something wrong. Okay, so in a single replacement reaction, we have an ionic compound reacting with an element. That element replaces something in the ionic compound to make a new one, and then whatever was replaced is by itself. If this is a metal, it replaces what in here? The metal. If it's a non-metal, it replaces the non-metal, right? That's all we have to remember. This replaces the thing it's like in the ionic compound. That's the only way you can get an ionic compound on the other side. Okay, could this also produce special elements? Yes, if it's replacing a non-metal, there's a possibility that you could get a special element by itself that you would need to put a 2 or a 4 or an 8 beside. All right, in a double replacement reaction, they're all ionic compounds. We switch the metals. Yeah, just swap the metals and you got 2 new ionic compounds on the other side. Balance the reaction. All right, ringing a bell. Okay. Okay, if we're predicting and balancing a chemical reaction, right? First thing we got to do is identify what kind of reaction is going on. Okay? Then we can figure out what the products will be. Then we swap and drop any ionic compounds. And we should also say, look for specials. Sounds like you're grocery shopping, but you know what you mean. Okay? Look for specials. Okay? And balance the reaction afterwards. Okay? After we've done this. If we don't do this first, are we ever going to get it to balance? Probably not. And if we do, it won't be right. Okay? So we got to make sure that we swap and drop and look for our special elements. Okay, so example number 1 here. I've got sodium reacting with magnesium chloride. What kind of reaction is that? Single replacement. Sodium, metal or non-metal? Metal. Who's it replacing? Okay, so over here then, I'm going to have sodium with chlorine, and I'm going to have magnesium by itself. All right, is magnesium a special element? No, it's not. So I don't need any numbers on that. Is this an ionic compound? What do I have to do with it? Drop and swap. Okay, +1, -1, is it good? Okay, now I can balance the whole reaction. What do I start with? Largest number. That's chlorine. Okay, there's 2 chlorine over here. Now there are 2 over there. How many sodium does that give me? Are we good? Okay. Second one. I've got diphosphorus trinitride. What kind of reaction starts with only one thing? Okay, so it's going to break down into its 2 elements. Right? Phosphorus comes as a 4, nitrogen comes as a 2. Okay, what do I balance with first? Biggest number. So phosphorus. There's 4 over here. What do I need here? 2. How many nitrogens does that give me? We good now? Okay. Looks like you guys are remembering this. That's good. Okay. What's the next one? Double replacement. Right. We got double replacement reaction. Okay. So who do I switch? Right. I switch the metals. So that means iron's new partner is chlorine. Hydrogen's new partner is oxygen. When hydrogen goes with oxygen, usually that's the stuff we make. Agreed? Okay. On this side here, I've got iron, but does iron have more than one possible charge? Okay, so how do I figure it out? Right, I look over here. What if iron was by itself over here? What if it was a single replacement reaction? Then what would I do? most common charge, right? Then I look at my periodic table and pick the first charge. Okay, in this case, I can figure out what iron is. Oxygen's always -2, so iron has to be 2 because they're 1 to 1 here. All right, so this is the 2 plus iron. Chlorine's always -1, so I got FeCl2 and H2O. So I can start with either chlorine or hydrogen. It doesn't matter which because they're together over here. And putting a 2 there fixes them both. Okay. All right. Reaction is combustion. What are always the products? All right. Do I need to apply the rule of 2? Okay. How many carbon do I have? How many hydrogen do I have? 20. Now, on here, I have 16 oxygen here, 8 times 2, plus 10 more is 26. What times 2 will give me 26? We only did that one about 500,000 times in the chem unit. Okay? Everybody with me there? All right. Okay, ringing a bell? De mole. Now we got to do de mole. Okay? Guys, you look at the mole equation now, N equals little m over big M. After all the formulas we manipulated in physics, that one's pretty simple now. Okay? It was a total nightmare for you when we did it back in chemistry, but now it's a cakewalk. All right? So if I'm going to find or do a mole equation, one of the first things I'm going to have to do is calculate the molar mass of the material that I was given. Probably on the test, I would give you just the name. So I wouldn't give you the formula like I did there. I'd say, what's the mass of X many molecules of calcium chloride, right? So what do you have to do first? Yeah, first I got to get this, right? Right? I've got to drop and swap it correctly. That got a lot of us on the unit exam, okay? I had 2 in there that you had to drop and swap, and people forgot to do it, okay? They would have written it like this and calculated the molar mass with only 1 chlorine when there are actually 2. Okay? You got to remember that when we have ionic compounds, we got to drop and swap. Okay? All right, so now I've got to drop and swap. Now I can do the calculation. So calcium is 40.08 according to my periodic table, and chlorine is 35.45, but there's how many of them? Right, times 2. So now I add those together. I think that's right, 110.98 grams per mole. Okay, we did water is the example right here. Okay, there's 2 hydrogens, so the molar mass of hydrogen is 1.01, but I multiply it by 2, and then I add it to the molar mass of 1 oxygen, 16, and I get 18.02. Is that ringing a bell for people? Okay, that's kind of the easy part of the mole equation, is just calculating that off the periodic table. Sadie. Yes. No different mass. Right, yes. There's only one. Yeah. And it's the same periodic table you had in the chem unit. I just shrunk it so it's not huge. Okay, it fits on an 8.5 by 11 sheet now. Right. All right. Other things, okay? Av's number, okay? What does Avogadro's number tell us? It's the number of other way around. Number of molecules in 1 mole. Okay? Doesn't matter what it is, this is the number of molecules in a mole. That's why everything has a different molar mass, because everything has different molecules, right? So we got to remember that that's what that's for. All right, let's try this example here. Calculate the mass of 8.05 times 10 to the 24 molecules of dinitrogen tribromide. That's as hard as they get. That's why I put it in there as the example. Now, do you have the ability to see 12 different examples somewhere? Where? Yes, because every day we did this in class, I did one example of each of the 4 ways that I could do this. Okay, so then we did that for 3 days. That's 12 examples. Okay, and just watch the podcasts for the mole equation. Okay, and I did that every day. All right, so what we know is the number of molecules and we know the material. If I know the material, what should I calculate right away? Molar mass, which is big M, right? Big M is molar mass. Okay, nitrogen is 14.01 grams per mole, and how many are there? Okay. And bromine is 79.90, and there are 3 of them. Okay, so 28.02, that's my nitrogens, plus 79.9 times 3. Yes, I did. 14.01 times 2 is 28.02. Yes, because 14.01 times 2. It's okay. All right, 267.72 grams per mole. N is the number of moles, big M is molar mass, little m is mass. Okay, what are the units for mass in chemistry? Grams, okay? I mean, if this is in grams per mole, this has got to be in grams, okay? All right, so I've got big M, and I've got the number of molecules. I need N still if I'm going to calculate mass. How do I get N with this number here? I divide by Avogadro's number, right? Okay, so N is going to be the number of molecules I have, 8.05 times 10 to the 24, divided by the number of molecules in 1 mole, Avogadro's number. So in brackets, 8.05 E24, divided by Avogadro's number, 6.02 E23. All right, so we get 13.37 moles. All right, now I have N and I have big M. How do I get little m? And how do I manipulate? What do I do with big M? Right, multiply both sides. Okay, so M equals little m, or little n, sorry, times big M. So that's going to be 13.37 times 267.72. Okay, that was our molar mass. That was our number of moles. All right, 3,579.98 grams. Okay, would be our final answer there. 2 decimal places is all you need. That ringing a bell? Okay, that's definitely something that would probably be useful to review a few times. Okay, there are 2 of those on the final exam. No, I don't have any numeric response. I want to give you as many marks as possible, so I want you to show work so that I can do that. Okay. Okay, and then you also have your solubility chart. Now, yours looks a little bit different than this, but it works still the same way. Okay, if I ask you, is this material soluble? So this is lead 2 bromide. Okay, first I look up the non-metal, bromine. Okay, if it's in the first row, it's soluble, but bromine is not soluble with lead. That's why lead is in the second row. Everyone remembering how that works? You'll have when you get your test, you'll have the periodic table, and you flip it over, the solubility chart will be on the back, along with like givens and formulas and stuff like that. It'll all be there. So it's kind of a separate sheet. I probably will have a couple of multiple choice that involve that. On your unit exam, I think the question went something like this. In this chemical reaction where, these 2 things react, which of the products will be soluble? And then it was, this, both, neither, okay, something like that. And then you have to use your solubility chart to figure it out. Probably something like that, I would think. Eli, put the phone away. Okay. Everyone all right with the solubility chart? Okay. Other things we went over in chemistry, we talked about polar nature of water. Okay, there could be some questions about that. Mostly that's in the biology part of the test, though, where we talk about plant transport, but it was still something we talked about when we talked about solubility. And then obviously the types of solutions could be multiple choice items, homogeneous, heterogeneous, things like that, solute, solvent, just terminology kind of stuff. Okay. Yes, good question. How do we do, because on your unit exam, there was a question that asked you draw a picture of an atom of, I can't remember what it was, but, and you had to get the right number of protons, neutrons, and electrons, and you had to draw the right model. Well, we would draw the cloud model because that's the one we accept. But how do I figure out how many protons there are? It's the atomic number. Okay, so number of protons equals the atomic number. What about electrons? Same, yeah. Okay. Number of protons and number of electrons has to be equal so that the atom is electrically neutral. All right. How about number of neutrons? Right. Atomic mass minus atomic number. Okay. So if we're looking at, let's pick one that's tricky. Okay. Magnesium, for example. Okay. So magnesium is 24.31 for its atomic mass. Okay, and its atomic number is 12. Okay, so I get 12.31. How many neutrons are in magnesium? 12. You can't have 0.31 neutrons. Okay, you can't have a third of a neutron hanging around. It just doesn't work that way. All right, always remember nearest whole number. I'll probably pick one a little more obvious. Okay, but don't write 12.01 neutrons because you can't have a fraction of a neutron. Okay. All right, good question on that one. Maybe. I'll give you a lot of details, but I'm going to tell you everything. All right. Any other questions on chem? Okay. So for bio, we started out with the cell theory. Yeah. Okay. We started out with the cell theory. Again, I'm not going to have you write these out verbatim in the written response, be able to recognize them. Okay. But the most important one is number 2. Okay, because this came back several times throughout the unit. Right? When we talked about the cell anatomy lab, it was part of our hypothesis. Okay, if cells have to carry out the basic functions of an organism, then they need to have these particular structures because they do these jobs. Okay, so that's something you might even be able to work into several of your written response answers. Right? If it's talking about cellular structure or modes of nutrition, things like that, you can probably work that in there somehow. Okay. So all organisms have cells. Cells divide to reproduce. Yeah, I could have a multiple choice question that asked you something like that, but mostly this is the one that's going to be most important for us. I'm probably not going to ask you about the 2 Swedish guys who came up with it, but okay, it was Schleiden and Schwann. Okay. All right, so that's really all you need to know for cell theory. Okay. Cell anatomy, on the other hand, there's quite a bit to do with that. There is a picture of a cell in the final exam. It's in the multiple choice this time, not the written. So it could be things like, what is the function of structure number 5? What is structure number 2? Okay, so you need to know names and functions of all the stuff in either a plant or animal cell, because I'm obviously not going to tell you which one is on there. Okay, only one is on there, but you don't know which, so you got to study them both. Okay. Other things, modes of nutrition. Okay, photoautotrophs and chemoheterotrophs. Remember, photoautotrophs can make their own food using light and carbon dioxide and water. Okay, animals have to consume other organisms. Chemoheterotrophs have to consume other organisms for food. That means they have differences within their cellular anatomy. Photoautotrophs need to have chloroplasts because that's what carries out photosynthesis. They got to have water vacuoles to help support the cells and the overall structure of the plant. Okay, and they got to have cell walls for the same reason. They've got to be rigid and support the structure of the cell and plant overall. Okay, if you're a chemoheterotroph, you have lysosomes because they digest food. You have centrioles, which help with cell division. Okay? And you don't have a cell wall, because you have to be flexible and be able to go out and get food. Okay, things they have in common. All of those. Okay, and remember, you only need to know kind of enough to recognize the function of each structure. Right? You're not writing out a detailed description paragraph of what the mitochondria does. Okay, you just need to tell me things like, oh, cell membrane controls not entry and exit, but endo and exocytosis. Nucleus stores DNA. Nucleolus makes ribosomes and RNA. Smooth ER, lipids, detoxify poisons, break down complex carbohydrates, make hormones. Rough ER, transport, put all that stuff. It's all in your notes already. You don't need to write it down again, but you do certainly need to go over it. Okay, there's several marks in the multiple choice to do with cell structure. Probably if there's one topic worth more marks than anything else in the multiple choice, it's that. Okay, because there's lots of structures within a cell. So I would certainly know that one. Okay, questions on that? Dan? No. I write the ones for my classes. Mr. Van Erf writes the ones for his classes, but we do sit down and look them over because we want to make sure they're similar level of difficulty. And we did, and they do, or they are the same level of difficulty. We do test the same things, and where our weightings of everything are pretty much the same. There's always some. I tend to emphasize physics a little more. Mr. Van Erf tends to emphasize biology a little more. Just he's a bio guy, I'm a physics guy. It just kind of happens that way. But in general, they were like statistically the same. What's different is you guys are familiar with the way I write my questions. So it's better to have me write your test and you might write Mr. Van Erf's test, because his test reads a lot different than mine just because of the way he words things, right? So we don't like to put together a common final exam and then you're going, that question doesn't make any sense just because you've never read a question made by Mr. Van Erf or Mr. Hesch or whatever. Yeah, so it's all mine. But you can rest assured that they are very similar in terms of level of difficulty. No one class is getting an advantage over another. You can. I don't know when you're going to get a chance to, because it's one of the last exams. But yeah, I mean, I have to keep them on file for, I don't know, I don't remember how many years I have to keep them, but I do have to keep them on file. So if you wanted to come and look at them, you could. Yeah. Okay. Yep. All right. So that's the stuff to do with cell anatomy there. Okay. And then transport within the cell. Was that a big topic? Huge. Okay, now, it was a huge topic. These three were not all that big. Phagocytosis, pinocytosis, and receptor-mediated endocytosis were minor, okay, very minor. In fact, if there's one question on the test that talks about those, it would be in the multiple choice. That's how small and minor they are, okay? The big three are those ones: active transport, osmosis, and diffusion. You had 4 or 5 questions on your written response part of your biology exam that was on this topic. And you had another couple that you could work it into. Okay, so it's huge. So we need to know the differences. Okay, we need to know the characteristics of each one. Active transport is unique because it uses energy and it can go against the gradient. Okay, it's across a membrane as well. Okay. And remember that this works because there are little tiny transport proteins that open and close to allow that pumping action to occur. Okay? So if something is talking about a situation where you're going to have more inside than outside, that's going to be active transport, or more outside than inside. Anywhere where it's not trying to equalize, that's going to be active transport. Okay? Osmosis, remember, is the movement of water across a membrane to balance what? Salts. The question on your unit exam was about the slugs who went in the Great Salt Lake. Okay, so being able to explain what osmosis is and what's going to happen as a result, that's certainly going to be important on your final exam. Okay, is osmosis also part of plant transport? Yeah, so osmosis was probably, the second most important, because diffusion was the most. Okay, huge deal here, because diffusion is the movement of everything. Not across a membrane, but within the cell, everything moves by diffusion from high concentration to low concentration. What were the drawbacks of diffusion? It's not targeted and it's slow, especially over large distances. Okay, this is the thing that worked back into surface area and volume. Why cells can't get really big is because of diffusion. They're transporting everything by this method, and it doesn't work well over great distances. Okay, this was also something you could work in if you were talking about an advantage multicellular organisms had over single-celled organisms. Multicellular organisms can get bigger because they're made up of small cells that overcome the problem of surface area to volume. Everybody follow me there? Okay, so those are all kind of things that those could get worked into. Cellular transport could get worked into several written response items on the final exam. All right, surface area to volume. You had the question on your unit exam that was the Starship Enterprise being attacked by the giant amoeba. Okay, why that couldn't happen? Because amoeba is a single cell. It cannot get that big because of diffusion. Okay, so diffusion's too slow, doesn't work across large distances, it's not targeted, so it limits cell size. Okay, cells want large surface areas and small volumes. That way they got lots of membrane to absorb stuff and very small distances to move it. Okay, so expect there's probably going to be something on there in either the multiple choice or the written, maybe both, that'll be on that topic. Okay, and then transport in plants. Okay, you got to know the parts of the plant. Okay, so that's xylem transports water, and that's going to use polar nature of water, so cohesion and adhesion to hydrogen bonds. Okay, osmotic pulling action, when water evaporates through the stomates, it leaves what behind? Salts. Yeah, there's salts in the leaf. That makes water move up into the leaf by osmosis in order to balance that. Okay, it's also that same osmotic process that can open and close the stomates. Remember that stomates are open, okay, when there's lots of water. But throughout the day, as water evaporates from the leaf, salt builds up in the leaf, and that draws water out of the guard cells of the stomate, and that makes them close. Okay, so again, it's cellular transport allowing a multicellular organism to do something important. Okay, again, working all of those ideas in together is going to be important. Okay, do you need to know the layers of the leaf? Yes, you do. Okay, because that was our example of specialization in a multicellular organism. Remembering that, of course, a multicellular organism has specialization, different cells for different jobs. Okay, and that's again another advantage they have over single-celled organisms. Okay, the examples of that: stomates, specialized cells that open and close. Okay, the spongy layer holding the water and allowing water to evaporate out the stomates. Palisade layer, all those cells packed in really tight to carry out photosynthesis. Yeah, definitely go over that. That's your example of a multicellular organism. Okay. Everyone all right with that? Okay, and then further about plants, plant growth. Okay, we talked about several hormones. We talked about several tropisms that plants employ. Okay, so tropism, that's when they change their shape, kind of get weird shapes or bends in them. Okay, phototropism, growing towards the light. Geotropism, growing up against gravity. Thigmotropism, touch, usually grabbing onto something and using it for support. Okay, the other thing we got to remember is that these tropisms, these shape changes happen because of hormones, okay? Auxins in particular, and that was the thing people forgot on the unit exam, right? Okay, on the unit exam we said, explain how this plant is doing this, and people said, it's growing towards the light, which is like 2 out of 5, okay? Probably was less than that. What we need to say is, when it bends, it makes auxins go to one side, that side grows faster, okay? That's what causes the bend. They call it gravitotropism. Yeah, that's the other name for it. Yeah, it's my test. Yeah, my test. Just worry about how I said it. Mr. Venner, who teaches bio, says it my way. Yeah, or as I like to think of it, the right way. Okay. All right, and then remember that these were the hormones. Okay, gibberellins, not a really important one. Abscisic acid, that was pretty important because what does it cause a plant to do? Not so much die, but go dormant, right? Okay, auxins. Okay, auxins are the ones that make it grow longer. Okay, that's the one that's big for tropisms. Cytokinins make the cells reproduce, divide. Cytokinesis is cell division. Okay, and ethylene makes fruit do what? Ripen, and plants often to die. Okay, so those are all pretty important. There's lots of diagrams on the final exam in the biology section, and it's photocopied in color. It turned out really nice. Okay, the pictures are all very clear and helpful on the questions, so make sure you look at them. All right, questions on plant growth and tropisms? Okay. That'll be in both parts. Okay, and that's it. Okay. We'll talk about Unit 3 and Unit 4. Okay, we'll do the review of that tomorrow, but I'll answer questions on the first two here. Yeah, I mean, roots are part of a plant. We need to know the functions of the parts of the plant. I only mentioned phloem and xylem, but yeah, roots and leaves are also important. Hey, what else, guys? Dave?
You said you'd have more application kind of questions. Would there be more of that on the final exam, or kind of?
For the biology unit, not as much. For the global systems unit, yes. There's going to be more application stuff for Unit 4. Unit 2 is going to be explain this stuff. Yeah, I mean, it might be in a real world situation, but it's still basically, okay, I remember how that works. In this situation, it'll work like this. Yeah, I did. 50 multiple choice. And I think I said 15 written. I'll just double check here. Okay, so there's 50 multiple choice, 50. Okay, and there's 87 marks in the written response. Okay, now some of that is like quick stuff, like balancing a reaction or something like that. Like it'll be quick stuff. Sorry, computer's locked up here, guys. 15. There's 15 written response items. Okay, but some of them are quick. Like I said, they're worth marks, but they're quick. Okay. Now, your unit exams, they've usually been around, between 30 and 40 multiple choice. Your last one was a bit shorter, but they're usually between 30 and 40 multiple choice, and they're usually around 8 or 9 written. Well, this is 1.5 times as long as a unit exam, because you have 1.5 times as long to write it. Okay, so it works out. No, I think I said it was 20, didn't I? Double check that too. Right, write it as a formula. So lead is Pb, and it's telling me it's the 4 plus kind of Pb, and it's with nitride, which is -3. So I'm going to Pb3 N4. I don't even know if that's a real compound, but we'll just say it is. Okay, so that's what we've got. Now I can calculate big M. All right, so for big M, lead is really heavy, 207.2. Okay, and there's 3 of them, so I got to multiply it by 3. And then I'm going to add that to the nitrogen. There's 4 of those. All right, 677.64 grams per mole. Okay, is our molar mass. And they tell me 16.5 moles. Which part of the mole equation is that? N, right. Okay, so N equals little m over big M. I'm solving for little m. What do I have to do with big M? Multiply it over here. Okay, so that's going to mean 16.5 times 200 or 600, sorry, and 77.64. 11,181.06 grams. Yep. I can have half of a mole, though. Remember, a mole is lots and lots of particles. Nope, there won't be. Okay. Other questions there, guys? All right. Then let's work on that review package for a bit, and then tomorrow we'll go over physics and global systems. Yep.